No, George. We'll go ahead and get started. Thanks everyone for coming to Dale Broder's PhD seminar. I'm going to tell you a little bit about Dale and where she comes from and her accomplishments. So Dale grew up in Alabama, as you know, and actually her parents are here. Oh, there they are, visiting from Alabama. Dale went to the University of Georgia for college, and she majored in ecology and German. And before coming here to CSU, she already had experience with various research projects, including working on fruit flies and dung flies at the University of Georgia, as well as at the University of Zurich in Switzerland. And as soon as she arrived here in our program at CSU, she became a really critical member of our large Guppy group. And I think the size of this audience is telling in that she's also been a really important influence on our community. And not only through her academic activities, but also her other activities. Some of you may know that Dale is an accomplished dancer. She dances in the community. She's also a singer. She loves to cook, and she's an all-around fun person who's just a pleasure to work with. In terms of her academic accomplishments, Dale's going to be talking to you today about her 4 chapters that make up her dissertation. But I want to emphasize that in addition to those 4 chapters, one of which is already published, she also was co-author or primary author on 8 other publications while she was here at CSU, with many others in progress. She also received the National Science Foundation Graduate Research Fellowship to support her work. So she's been very successful in her research, but she's also been successful in her science education work, which she has a real passion for. So I want to just highlight some of that. Dale's going to be talking a little bit at the end of her seminar about some of her outreach work. But I just wanted to emphasize that she created some really engaging activities to teach science and evolution to K-12 students. And she turned those into established regular programs that she led every year at a number of local schools, including a primarily Hispanic school in Greeley. And so some of you may have seen these very young students presenting their research at the Front Range Student Ecology Symposium each year, and that was thanks to Dale. And so her outreach activities have been highly successful, praised by teachers and principals, and even attracted some local media attention in the local papers. But I think the really cool thing about her outreach is that she took the initiative, along with folks in our Guppy group, including Cameron Gallimore and Emily Payne, and folks at the Education Outreach Center, to turn these activities into Guppy kits that can be used over the long term in local schools. So her outreach work will have a long-lasting legacy even after she leaves CSU. I also just want to emphasize her contributions to undergrad education here at CSU. So she was a TA for a number of courses. She also served as a primary instructor for animal behavior this past summer. And in terms of grad education, she actually helped to develop a graduate course when she recognized that communicating science to the public was something that was lacking in a grad program. And so she worked with Graham Peers to develop develop and co-teach graduate course on that topic. And then finally, I want to emphasize her really important, I know, I can't stop here, important contributions to undergraduate mentoring. So Dale won awards at the department and the college level for mentoring undergrads. She mentored over 20 students, and many of them worked with her over multiple semesters. So when you add up the number of semesters that all these students worked with her, she provided over 50 student semesters of mentorship to undergraduates. Many of these students worked on independent research projects that were separate from her own research. Over 15 of them presented their work at regional and national conferences. 5 completed honors theses working with her, and 3 submitted manuscripts for publication. So she essentially established this mini empire of undergraduates where she was another PI of a subset of my lab. So this day, I think, because she's contributed so much to my lab and the department and the community, is really bittersweet for me, emphasizing the bitter here. I'm so happy and proud of her accomplishments, but I'm going to be really sad to see her leave. Luckily, she's not going very far. She's just going down the road. She got a postdoc at the University of Denver, so that's exciting for all of us. So I'm now going to turn it over to her so she can talk to you about her dissertation work. Please join me in welcoming Dale Broder. These are switches. You know, they don't. Oh, yeah. All right. Is that too dark?
Yeah.
Yes?
It's 4 o'clock.
How about that?
Switch it in the back.
Oh, okay. All right. Thank you so much, Lisa, for that beautiful introduction, which means I have a lot to live up to in the next 40 minutes. But thank you guys so much for being here. It means so much to me that you all made it. So today I'm going to talk to you about some of the work that I've done here. In the beginning will be about evolution and plasticity, and the end will be about some education work. Okay. So one of the major questions in biology is how do organisms respond to changing environments? And so this could be over short time scales. You could think about everyday changes, over seasons like this wet and dry season, or over years or decades. And this question also has conservation implications because humans are changing the environment really rapidly. So animals have to respond very quickly often. You can think about climate change, habitat degradation, invasive species, or overharvesting. But when we think about how organisms respond to changing environments, there's some main ways that they do this. And the first is that they could evolve. And so when we talk about evolution, we're talking about genetic changes or changes in allele frequencies in a population over many generations. So here's a classic example of a pepper moth, which is blending into this tree, but after the Industrial Revolution, with all of the soot on the tree, there was selection for this darker, more generations. But there's also another way animals can respond, which might be more rapid, and that's plasticity. And this can happen in an individual's lifetime. And so these are non-genetic changes in response to the environment. So for example, these are actually water fleas, or Daphnia, and they're actually clones, so they're genetically identical, but one that lives without fish has this phenotype, this is how it looks. And then the one that lives with fish predators develops this armor. And so we know that these processes work together to shape the phenotype, or what an animal is, what it looks like, how it behaves, all the traits about an organism. That's shaped by their genetic contributions as well as the environments that they experience. And so the relationship between evolution and plasticity is quite complex, and many have debated and discussed it, including Cameron Gallagher and a lot of other people at CSU think about this question a lot. But today I'm gonna try to explain some of the main points of the theory using this cartoon that I made. So you can imagine that we have an environment which ranges from blue to red, and in this bullseye or target is showing you the optimum. So let's say that the optimum phenotype is to background match. So you can see this star creature, it's pretty well matched, it's pretty close to the middle here. However, if the environment shifts, and now the new optimum is to have this red phenotype,
So in some ways, plasticity could help because it could allow organisms to maybe move part way and therefore not go extinct. And then maybe selection over many generations could bring that population to the new optimum. But it could also hinder evolution if it allows the organism to make it all the way to the new optimum, because in that way, it's essentially buffering or shielding evolution. And so to illustrate that idea, I'll just go back to this example. So if the target is an environment without fish predators, this would be the optimal phenotype. And then if in this alternative environment with predators, if every individual, regardless of their genetic background, if they can achieve the optimum, in this case growing armor, then selection wouldn't be able to act to allow these organisms to evolve. So that's kind of some of the theory. And to try to ask questions related to these ideas, we use the guppies, which you guys know that there's so many people in this department studying guppies. So for those of you who are already familiar, you can chime in and help me along here. So guppies are found in these really different environments in Trinidad. So we can find them in hibernation environments where they live with lots of predators like the cichlid here. But then as you move upstream of the river, you encounter these waterfall barriers which prevent predators from moving upstream. But the guppies have colonized these reaches multiple times, and there they've evolved with low predation risk. So we call these high and low predation fish. I think my mouse is like on the screen. Okay. And so the reason this is important is that the guppies that live in these different environments are really different when we think about the traits that they have. So you can clearly see that these are male guppies. You can see the one that lives with fewer predators is more brightly colored. And lots of traits vary across this continuum. So if you think about morphology, so I'll just show you a few examples. So for example, the fish in these high predation environments tend to be more streamlined. That's some of Corey's work. And then life history traits, timing of important life events. So for example, females that live with these predators might mature earlier but have smaller or fewer babies, for example. And of course, behavioral traits also vary. So regarding mating behavior, males that live with few predators tend to perform these showy courtship displays called sigmoid displays that are really conspicuous. And then in environments with many predators, the males use these forced copulation attempts that are sneaky and less conspicuous. And so we can use this system to really think about questions regarding how the genetic background in different environments work together to shape phenotypes and how it allows organisms to respond to changing environments. And so to do that, we can actually collect fish from these different environments, so from these low predation, high predation streams, and we can bring them into the laboratory, and we can actually rear them under the same conditions for several years. And in this way, we're controlling the environment, and then any differences we observe, we can attribute to genetic changes. Conversely, we can then take these same fish, and we can actually control the genetic background by using closely related individuals, in this case brothers, and then we can manipulate the rearing environment so that, in this case, one brother is exposed to normal water, and another brother is exposed to water that contains chemical cues of predation. And that way we can quantify the effect of the rearing environment on the phenotype. And so I've used this approach to tackle some fun questions, I think, in guppies, but today, in the interest of time, I'm just going to zoom in on the three sexiest topics. So cerebral laterality, genitalia, and mating behavior. So cerebral laterality, what is that? It's just partitioning of tasks between hemispheres of the brain. So you're probably familiar with left brain, left-handed, right-handed. You may not realize that this is pretty ubiquitous across animals. So guppies don't really use left and right hands, but we can actually look at their eye use. So guppies tend to use their right eye and the left hemisphere of their brain to view predators. And they use the left eye and the right hemisphere of the brain to process information about potential mates. And so, let me go back here. So when we think about laterality, we think about degree of laterality. And so what I mean by that is if an individual consistently always uses the right eye and the left hemisphere of the brain to view a predator every time, that individual is really compartmentalized. And so that would be highly lateralized versus using the different hemispheres half and half, for example. And what we know is that really highly lateralized individuals tend to outperform non-lateralized individuals in lots of tasks important for surviving with predators. For example, spatial orientation, shoaling, which is like schooling. They have faster escape from predators, and they're better at multitasking, like foraging in the presence of predators. So it would follow that predators should select for a higher degree of laterality. and that's what many people think. However, there is a lot of variation across populations, across individuals, in the degree of laterality that we see. And so a really interesting question is, what's shaping the patterns of this trait? And using the system, we're in a unique position to actually quantify the effect of the environment. And most studies aren't able to investigate this simultaneously. It's often ignored in this work. And so we can take populations from Trinidad, from these environments with and without predators, and so that would be these different genetic backgrounds. And then we can bring them to the lab and breed brothers in different treatment to find out some information about the effect of the environment. And so then we can perform these laterality assays. So here we place a guppy in this acclimation chamber. This is a goldfish. Pretend it's a guppy. So it enters this arena and it encounters the semi-opaque barrier, and this fish has to turn left or right to view this target. So this individual turns right, and it's using its left eye and the right hemisphere of its brain to process information about this target, which is a fish model. So we can assay an individual 10 times and calculate his laterality score. And so we did that and found, so here I'm going to show you those brothers breed without cues on the left and with cues on the right. And so actually what we found was that being reared in the presence or absence of these chemical cues was the most important. And in fact, we didn't find an effect of population or where these populations came from. So the A's here show that there's no difference between those two squares. So the fish who originated from streams with predators and those from streams with few predators didn't differ when they were reared without these chemical predator cues. But they all showed this increase in their laterality when they were reared with these cues. And so what that means is that we found a really important role for the environment in this trait. And we also found that it's in an adaptive direction. What I mean by that is that the plasticity is actually moving it towards this optimum. So when there are predators present, we know that a higher degree of laterality should be advantageous or confer some benefits. We see that that's the direction of the development of plasticity. Okay, let's talk about genitalia. So guppies give birth to live young, so they have to transfer sperm. They also have to transfer sperm to females, and they do that using a gonopodium, which is their intermittent organ. It's non-retractable. It's about 25% of their body length. So the cool thing about genitalia is that it's fascinated people forever. Darwin wrote 2 books about genitalia in barnacles. So for a long time, people have been really interested in genitalia because there's so much variation. So this is a really cool art exhibit. And so harvestmen or daddy longlegs are the only arachnid with a penis, actually. And so these are just some examples of penises of really closely related Apiliones harvestmen. And so people have often wondered what drives this rapid evolution or diversification of genitalia in animals. And in the guppy system, I'm just going to talk to you today about a couple hypotheses that are related to sexual selection. And so those are that females may prefer larger genitalia. So this is one. There's been some support for this idea that female guppies prefer longer gonopodia. But on the other hand, there's this alternative kind of prediction that actually longer gonopodia allow males to force populate females more effectively, and there's some support for that. So it could be more of a sexual conflict situation. And so since we're actually really interested in rates of evolution, we were actually able to take advantage of this experimental introduction in Trinidad. And so some colleagues actually moved guppies from a source population here, S, where these guppies are living with many predators, and moved them into these 4 replicate low predation strains with few predators. So those guppies are evolving for 1 to 2 years in this new environment. And then we can collect fish from these environments and bring them into the laboratory, into that same common environment. And then we can rear brothers in those 2 different conditions to manipulate that rearing environment. So using this approach, we can now think about overlaying those predictions on this system. And so we know that in these environments with reduced predation risk, that female preference is likely very important. So we would expect longer genitalia in those 4 introduction environments. But if this is supported, then we know that these types of mating strategies are really important in these environments with predators, and we would expect longer genitalia in these environments. So for my actual predictions, when I'm looking at those 4 introduced populations, if they become larger in those introduced populations, that supports this female preference idea. If they become smaller, that would support this sexual conflict. And so I'm going to show you what we found putting body length here on this axis, since body length tends to be highly correlated with conflict length. And so here you can see that the source population is the black line. And so for a given body size, so say 15 millimeters, for a given body size, all four of the introduced populations actually have shorter genitalia than the source population. And so what that means is that in only about 4 to 8 guppy generations, we see this repeated kind of parallel pattern of rapid evolution where all four of those populations involve smaller genitalia. We actually didn't find an effect of the rearing treatment. And so previous work that has compared the effect of predation on genitalia has found really mixed findings. So there's really no consensus. And ours is really the first study that's been able to actually isolate the genetic contribution to this trait. And we're able to show rapid genetic change in this trait in just 4 to 8 generations. And also we can say something about the direction. So the introduced populations all evolved smaller genitalia, which supports this sexual conflict hypothesis, which has been supported in other related fishes recently. All right, so now I'm going to talk about mating behavior, but before I get into it, I have to go back to this original conceptual framework of the star living in the blue and red world. And so I talked to you about how the star could match the background, and plasticity could potentially allow this individual to achieve this new optimal phenotype if the environment were to shift. But actually, it's possible that the environment could revert back, right, within an organism's lifetime. And in that case, depending on the type of plasticity, the organism may or may not be able to track that change. And so what I mean by that is, if you remember the example I showed you earlier of the water flea, and when they're reared with fish predators, they develop the armor. So if the environment reverted back, it might not be possible to ungrow the armor. And so when we think about developmental plasticity, we often think of it as being irreversible or fixed after a certain point in development. Whereas there's a different type of plasticity called contextual plasticity, where organisms can actually be flexible within a given situation or context, and that would allow them to perfectly track an environment in a reversible way. And so we call this contextual plasticity, and it's actually really important to consider when we think about behavior, because we think that behavior is particularly contextually plastic, but it may also be developmentally plastic. And studies that actually look at both types of plasticity and evolution in the same study are currently lacking. And that's what we hope to do with these mating behavior experiments. And so we use that same framework where we use this experimental introduction, and we're going to be comparing the source population with those 4 introduced populations, rear them in a common lab environment, and allow brothers to be reared these different rearing treatments. And so we conduct mating experiments with and without competition. So that's 1 male and 1 receptive female, or 2 males and 2 receptive females in the case of with a competing male. And for today, I'm only going to focus on 2 mating behaviors, those ones we've spoken about earlier, those showing courtship displays, the signaling displays, and the forced population attempts. So we think of these as alternative mating tactics. But wait, what about contextual plasticity? I told you that was important. And so the way we're going to measure that is within each mating experiment, we expose each individual male to an observation period either with or without alarm cue. And that alarm cue comes from other guppies. So when a guppy is eaten, chemical cues come out of the guppy's skin, which alert other guppies in the area that there's danger. So we used Bayesian bivariate MCMC GLM model. You don't have to pay attention to this, everyone. But yeah, so these data were percent distributed. And when I say bivariate, that means that both of those behaviors, the displays and the copulation tips, are both on the left side of the equation, since we think they're correlated with one another. And then I'm going to walk you through our predictions when I show you these model effects. So the first fixed effect in our model was population. And so that should help us answer the question about if evolution has occurred. So we can compare the source population to the 4 introduced populations. And then for the plasticity in our predictions. And so what we would expect here is those populations that have been evolving for 2 to 3 years with reduced predation risk, they should increase those showy courtship displays and decrease the forced population attempt rate. And then for those treatments, so for the 2 types of plasticity, The rearing treatment is exposure to these chemical cues throughout development, and then the contextual plasticity is exposure to these cues during the mating trial. And in both of those cases, with the presence of this chemical cue of predation, we would expect a decrease in those showy displays and an increase in forced copulation attempts. We had random effects because we had measurements on individuals more than once. We had brothers represented across rearing treatments and a family effect. And in the trial with competition, we have those two males are not independent from one another. Okay, so what did we find? So first, developmental plasticity. I won't show you any figures because we actually found no evidence that there was any effect of being reared in these different environments, in these mating behaviors. For contextual plasticity, so for this one, we expected a reduction in these showy courtship displays when the alarm cue was present in the experiment. Also, let me just explain that on the axis here, I'm actually showing you the raw data because the model is on a log scale, so it's not as easy to interpret. But this would show that males were performing here 1 sigmoid per 5-minute observation period. And so we actually found that without competition, just with one male and one receptive female, we saw the pattern we expected, this reduction in display rate. We actually saw the opposite pattern when there was a competing male. And we know that a competing male can cause guppies to increase their display rate, but it's interesting that this potentially maladaptive response was kind of trumping the natural contextually plastic response. So for the forced copulation attempts, we expected an increase when the cue was present here. And we actually saw a decrease both with and without a competing male. But this could be adaptive. This maybe shouldn't be surprising because we do know that a general reduction in activity is often a strategy used when predators are present. And so now thinking about evolution, these are going to be population differences. And so here we're comparing all 4 introduced populations to that source population. And so for sigmoid display rate, we expected them to increase their rate of sigmoid display in this environment with reduced predation risk. And instead, we see really mixed results. So we have one population, you can see over here, intro 1, which reduces their rate, and intro 4 increases their rate, and 2 others don't diverge from the source. So we don't actually see repeated parallel patterns in all 4 populations like we did with the gonopodium results. Similarly, for the number of forced population attempts, we expected a reduction compared to that source population, and we again don't really see repeated patterns. We see a couple cases where they actually move in the opposite direction than our predictions. I think it's interesting to note that with a competitor, the magnitude of the number of behaviors performed seems to be higher. So it seems to be a pretty interesting effect of having a competitor there. And so just to wrap up, we didn't find any evidence of developmental plasticity, but instead we found a really important role for contextual plasticity. And so for all populations in all rearing treatments, there is this general reduction in mating behavior, except for this case where a competitor was present. And so we actually did see some evidence of evolution in only 8 to 12 guppy generations, but it wasn't consistent across populations or even in the direction. So at the very beginning of this talk, I spoke about the relationship between plasticity and evolution. So after all of this, can I even say anything about that? So kind of. So I'll kind of just wrap up with all of the traits that we looked at. And so first, when we looked at cerebral laterality, we found a really important role for the rearing environment in shaping that trait. When we looked at genitalia, we found a really important role for genes in shaping that trait. And we actually saw rapid evolution in the absence of plasticity. And then for behavior, we actually had to change the diagram because now we have 2 different types of environment, this rearing environment and also the immediate context. And we found a really important role for the immediate context and the flexibility of those behaviors. And we did find evidence of rapid evolution of these behavioral traits, but it wasn't consistent. And so really we found everything, right? We found cases where the environment was important, where the genes were very important, and where both were quite important. And this just definitely indicates that it's a complex relationship that's likely trait dependent, and that it would be important in future work to take a more holistic look, since ultimately if we care about predicting how animals will respond to changing environments, we need to take a whole organism perspective. So now I'm going to change gears. I'm going to talk to you about education. And so everyone always has a diversity slide, and so I decided to have one. And so here's a slide that shows the diversity of life. These are all my favorite animals. And so evolution is the best explanation we have for the diversity of life on this planet. And it's incredibly important that we use evolutionary approaches to tackle some of the world's pressing problems, like climate change, biodiversity, curing and preventing diseases, etc. But despite that, the majority of the US rejects evolution. So this is a huge problem. And I argue that one of the big challenges when we try to teach evolution is that we don't teach it until middle school in the US. And most middle schoolers already have preconceptions about evolution. And we know that having preset beliefs really biases and impedes learning. There is some hope. So there are some things that have been shown to help overcome belief persistence, like maximizing student engagement, or by exposing students to causal evidence, or by holding them accountable for their conclusions that they make based on that evidence. And so in looking at this list, it was clear to me that these are all things inherent in the scientific process. Because when we do science, we're engaged, we're collecting causal evidence, and we're making conclusions, and we're held accountable for those conclusions to our peers. And so I was excited to discover that there's a teaching approach called authentic science, where you teach by letting students behave as if they are practicing scientists. And so I wanted to try to use this approach to teach evolution. And many have argued that inquiry approaches should be really effective for teaching evolution, specifically authentic science approaches. And some programs have been developed, but very few, because it's quite difficult to allow students to observe evolution in a typical one-week lesson. And so for this work, our goal was to try to teach evolution by natural selection. But we really wanted to use live animals, so that's been shown to be particularly engaging. We really want to engage the students. We wanted to use an authentic science approach. We wanted to show if it worked. And so we did pre and post assessments for knowledge and acceptance of evolution. So I needed some collaborators to do this. And so I worked with Severance and Windsor Middle Schools and all of these really fantastic 7th grade teachers to put together this program. So it took a year to develop, and we implemented it for 2 years. And now I'm just going to walk you through what we did for the program. And it should look a lot like the scientific process for those of you that remember what that looks like. So first, you just observe animals in this case. So we brought guppies to the classrooms. We had 3 different populations. And you guys should be familiar with the one on the left and the one in the middle. But we let the students observe these guppies and ask questions and make predictions to try to explain the differences they observed. And we spoke about these different places you might find guppies, with many predators, with few predators, and also domesticated. So these are the same species that can still breed with guppies, but they've been domesticated for hundreds of years. And we don't tell them which population corresponds to which environment. And so then the fun part is they get to do experiments. And so they get to help design and conduct these experiments, in this case, observing guppy mating behavior. And so they did the exact same experiments that I did for my dissertation work. They watched 1 male and 1 female. I recorded numbers of forced copulation attempts, number of sigmoid displays. They called it going for it. instead of forced copulation. Same idea. And then we also did some experiments with predators. So here you can see a cichlid predator here, and my partner Ryan here, Cameron Gallimore, who was integral in carrying out or implementing this program. You can see there's, you may be able to see 3 male guppies over there, so 1 male from each population, and the students made predictions about anti-predator behavior as well as the predator behavior. And then, of course, they enter and analyze their results. So then, on the last day of this week-long program, our goal was to bring this all together and actually try to teach evolution. And we used the mnemonic VIST, and I'm going to just walk you through these main ideas which are important to understand, to be able to understand evolution by natural selection. And the first is that variation exists. There's variation within and among populations, and the students can look at their data and see that quite clearly. This is just some example plots the students made. So you can see individual males within a population are really variable in their mating behavior, and then among populations we see a lot of variation. The next big idea is that much of that variation is inherited through genes. And we do kind of square activities to think about how traits are passed on to the next generation. but not all individuals get to survive and reproduce because of selection. And so they can clearly think about survival by looking at their results from the predator encounter. And so this is data from one school where the domestic guppy was eaten 6 out of 6 times. So you can see that this is not a random process, right? And they also were able to think about sexual selection because they conducted those mating experiments, and also artificial selection. We'll talk about that in the context of the domestic guppies. And finally, when all of these things are happening over time, that's going to lead to changes in allele frequencies. And to get this point across, we use spot exercises where we imagine that big A stands for a brightly colored male, little a would be a dull male, and how in various scenarios, for example, if there's a predator there, what might happen to the ratio of little a's, big A's. And at the end of this spot exercise, we say the E word for the first time, that a change in allele frequency in a population over time is called evolution. So we did this program for 2 years, and in the second year, we gave pre and post assessments, a 7-question multiple choice test to assess knowledge, and we made instruments to assess acceptance of evolution. And we had 204 students that had both a pre and a post test in the second year, so it was with an individual. And then we had 74 students who learned the same material at the same time using hands-on activities but just didn't participate in our program. So we call this a control group. Okay, so what do we find? So you can see that in the knowledge test score, which they could score up to 100%, so there's no difference at the beginning, but after participating in our program, you can see that our participants really increased their knowledge, and the control group didn't change. We saw a similar pattern for acceptance, where they diverged, and our participants had an increase in their acceptance of evolution. So we feel that this approach of using authentic science and live animals is a really effective way to teach evolution. And we were so excited about this program that we wanted everyone to be able to use it. And so for the past year, I've actually worked really closely with Emily Kane, my partner in crime, as well as Andrew Warnock, who heads up the College of Natural Sciences Education and Outreach Center. We actually packaged this whole program into a kit, and students can work through this self-guided kit and discover evolution by natural selection independently. And for those of you who are not aware, this is a fantastic resource, the College of Natural Sciences Education and Outreach Center. And so they're maintaining our kits, and they'll be using them all throughout Northern Colorado indefinitely, even after we leave. And so they are really looking for other professors and grad students to build kits with. All right, so this is the end. Back to the title slide. So basically, by using this natural introduction in the field and coupling that with this rearing environment in the laboratory, we're able to ask some really important questions about the relationship between plasticity and evolution for traits that are really interesting. And we also are able to use this charismatic system to create an educational tool. And so this brings me to acknowledgements. So there's a lot of them. So first off, funding was from the National Science Foundation and the Society for the Study of Evolution funded some of the education work. So also, I want to just thank CSU. I feel so lucky that I ended up here. The biology department, GDPE, this entire community is just fantastic. Like the amount of support that I've gotten and speaking to friends who didn't end up coming to school here, it's just unbelievable and really, I think, atypical. And so thanks for that. Okay, so all of this work has co-authors, and so these are all co-authors in order of appearance. And so thanks to all of these people who really made this work possible, especially a lot of the ideas of plasticity and evolution. Cameron was really important in helping me think about that. Lisa, my advisor, I'm going to thank Lisa for being such a fantastic advisor. I couldn't have asked for a better advisor. She's brilliant and was incredibly supportive, and also was a great role model. So she's really successful, but also has a great balance and has a really wonderful life as well. I'm getting pushed. So you're supposed to embarrass your advisor now, but those of you who know Lisa know that that's not possible. She's so poised and professional, there are no embarrassing photos of her out there. But I did find one that I think is representative of how supportive she's been. And so my my mom made these terrible headbands at my Lisa wore these cheesy headbands with so much pride and enthusiasm, and that's the same kind of support and enthusiasm that she showed me when I suggested, what about if most of my dissertation is about education? She was like, great, let's do it. And so I think that's really valuable. I also want to thank Mentor, Guardianship, Cameron, and my other committee members, Kim and George, who are being incredibly supportive, also encouraging me to pursue this education interest. Drew Denog has been a great person to talk animal behavior about. Graham helped me develop communication course, which is awesome. And Janice Moore has been a teaching mentor. Undergrads. Lisa mentioned that I worked with a lot of undergrads, and I definitely couldn't have collected all of these data without the help of this army. And so these students, were integral for this work, and also their enthusiasm and passion and creativity was really inspiring. Okay, the CSU support crew, many of you in this room have provided fantastic feedback over the years and just been really supportive. So thank you all very much. Friends and family, I have a great network of friends, so thanks so much to all of you, especially those of you that could make it today, and those of you that are streaming. And then especially, I just want to thank all the local comments. So all the local comedians, you guys have been a great network, most importantly because you make me feel better about myself. Because of your great self-deprecating humor. I want to thank my dance family, they're beautiful and supportive and amazing, and my immediate family, which I think I won the family lottery. I really lucked out, and thank you guys so much for supporting me through all this. All right, we do have time for questions. Mark.
Yeah, the chemical cues used to simulate predation, was it literally just drop dead guppies in it?
That's a good question. Yeah. So for the chemical cues that they experience throughout development, actually Corey and a lot of people developed this really cool system where you can place a predator in the sump of the system and feed that predator guppies, and that water just gets pumped through all the guppy tanks. And then for the actual smell of death from the guppies, it's in their epidermis and their skin cells, and so you can actually euthanize them and lacerate the epidermis to get this chemical cue extracted and then mix it with a solution of water and drop it in. Yeah. Brett.
What's an acceptance score? What is that? What's the value? It's like 3.
Yeah, sorry. Yeah, that was a Likert scale, so to go from 1 to 5. So it's like 3.5.
And so then they still had, not everyone accepted evolution. I assume that's what the acceptance was, right?
Right, and so it's a series of questions. I can't think of an example right now, but evolution is a great explanation for something, and then they can strongly disagree or strongly agree. And so 3 would be kind of neutral. So yeah, I guess they're around 3.5, which is maybe a little above neutral. That's a good.
So there's still a lot of work to be done.
Yeah. Yeah. Ellen.
I'm curious, you mentioned the E word, and coming out of this, if you were to do it again, and even in casual kind of interactions with people, would you pull that back still, or what's your perception on that, or introduce it earlier?
That's a good question. It just depends on how long I want to talk to that person. So in Alabama, I just say, I'm a scientist. Unless I just really want to talk for a long time. Out here, people are usually pretty interested in hearing about it, so I usually would say it up front. But as far as in teaching, I've spoken to a lot of teachers, so we've had a lot of teachers come try out these guppy kits, and we had some teachers that were 3rd grade teachers who are really interested in using components of this in their classroom. So I think the earlier you introduce it, the less scary the word is. Yeah.
Have you noticed any effect of being really nice to the guppies, telling them that you love them every day, so do good things?
No, I haven't actually. We try
control group was still taught about evolution, because not by your program, right? So how, can you kind of summarize how it was different?
Yeah, so I would say that this study is a first attempt, and so I would say this is still preliminary because it's not a perfect control. And so this teacher used a textbook, worksheets, video games, and also we weren't in the classroom, and so there were several factors that differed in that group. But she basically kind of just used traditional techniques and had the same teaching objectives and teaching goals at the same time.
But they still use the word evolution, right?
Yeah, they use the word evolution, and they use all the same terms, natural selection, artificial selection, they have the same teaching goals. So when we really are excited about assessing these kids, and we should be able to have a lot more control over the assessments that we do with these guppy kids to ask really specific questions. For example, kids have been shown to be really effective with girls, which is an at-risk group for participation in science. Luke.
Has anyone looked at the rate of evolution of the predator? Are they all in the same rate as the guppy?
That's a good
Is this
question.
like countermeasure to the?
That's a good question. You mean in their environments, in the wild,
Yeah.
or in the lab? Yeah, I guess I'm not quite sure what you're asking. Well, in the environments with predators as compared to ones with few predators.
Do the predators change their rate similar to guppies in terms of type of?
I don't know. That's a good question. So, I mean, that may be something to think about for novel predators. I don't know. So I know in Trinidad, tilapia farming is becoming a big deal, and tilapia are escaping, and so we have these novel cichlids in the environment. And so in that case, if they don't have experience with guppies, maybe they're evolving ways to be effective guppy predators or something. But for these kind of populations, which we kind of call hybridation, they've been kind of coexisting for so long that I don't know that people have looked at evolution. Yes.
So I saw in the control group, the second side of the control group, less kids believe in evolution after. Can you speculate as to why that would happen?
Yeah, that's a great question, actually. So we have a significant interaction, which means that those slopes are different. So one slope slightly increases and one slope slightly decreases, but I would actually have to do a different statistical test to say if the pre and post in the control is for sure different. They could be not significantly different from one another, that's possible. That's not the great answer.
Can you show that to yours?
Yeah.
It looks like it's within the margin here.
Yeah.
Yeah. So I would have to do another post hoc test, I think. But it's also repeated across individuals, so it's tough to say for just looking at the raw data without looking at a model.
All right, I think we have time for one more short question. Yeah.
I was just curious about maybe, like the home life of these kids, like whether that.
Yeah, well, certainly that's definitely been shown in other work that that's a huge component, actually. One thing I'm really excited about, hopefully going to do in my postdoc that I'm going to do, is we're going to be working with a lot of Hispanic communities where we're going to hopefully have kids that go home to the whole family, so the whole family can do kids together, which has been shown to be really effective for certain communities. So that's something I'm interested in exploring in the future for sure.
Oh, Mina wanted to ask one last
I just
question.
wanted to ask, we can't keep kids in the lab and near them, so we know that they have the same. So I'm just wondering, what measures do you use to ensure that your control students were actually, you're comparing apples and apples, so that you're just measuring the effect of the intervention?
Right, yeah, and so basically we did this program with the entire school, so it was just every single student, and it was just by chance, just 3 classes didn't participate and 12 classes participated. So there wasn't any
Random assignment.
it was just, yeah, so we just did the entire school both years, so it's like 600 students. And we didn't collect any information about students at all. So we don't know anything about them, we just have pre and post.
So you can't retroactive this in 20 years.
All right, please join me in thanking Dale.
Really